So welcome students, we will see now the biological hydrogen production in this video. So it is only remaining thing in module 3.1. After this the module 3 is over. So what is coming to the starting, basically what is the hydrogen? We come across with a very simple molecule, we come across routine discussions about it. It is a chemical element with symbol H and atomic number 1. So chemically, if you say what is hydrogen, which is the first entry in the periodic table due to atomic number 1, it has atomic weight close to 1, 1.00794 atomic units, and being this makes this as a lightest element with one proton, one neutron, and one electron. It's a monoatomic form, H1, not molecular, atomic form, is the most abundant chemical substance in the universe. So entire universe has 75% mass is H1. So that much abundantly found is the hydrogen in the universe. And standard STP, that is called as standard temperature and pressure conditions. Hydrogen is very fragile molecule. In terms, you cannot detect it simply because it's colorless, odorless, tasteless, non-toxic, not having any taste, non-metallic. Apart from this, it has very highly combustible and diatomic gas. So this particular combination is very dangerous for hydrogen. And so usually for handling hydrogen, you have to take extreme safety conditions, safety measures due to its inherent high combustible state combined with difficulties in detecting hydrogen in air. So it is very difficult to detect leakage of hydrogen as it is very colorless, odorless, tasteless. It's non-toxic, non-metallic. Mostly it is non-reactive in sense you can't have any detecting type of test. It is highly combustible. So only detection is combustion, which is which can be used routinely. It's very dangerous, but at the same time, due to its combustible property, it has can be used as fuel. So there are basically two methods for biological hydrogen production: light-driven and dark fermentation. So in combination of light, when the sunlight or any other light is present, there is one particular pathway by which we can get hydrogen. Or in the dark, in absence of light or light independent process, it is a fermentative production. In light driven by hydrogen production, as it uses light, first is biophotolysis, which is nothing but a usage of plant cells to break molecules in water or any other hydrogen containing substrate to generate hydrogen out of it by catalysis. And as we are in the microbial bioengineering, it is this part of the hydrogen production is out of our syllabus scope. We are dealing with photofermentation using bacterial photosynthesis, not plant-based photosynthesis. So in bacterial photosynthesis, we use microorganisms such a way that they should be able to capture solar energy and carry out the conversion of substrate to hydrogen. So they are being investigated as a part of two-stage systems. One can be used as a light-based system along with dark and hydrogen producing bioreactor. This step requires ATP acquiring nitrogenase enzyme. It reduces proton to hydrogen in absence of other substrate. It is called as photofermentation. How to improve this photofermentation is the reduced pigment mutant of rhodobacter. It is a rhodobacter is used for photofermentation. It can be used as it reported 10 watts per meter square. Very low light intensities. It gives higher hydrogen production. But the difference in higher electricity, that is 100 watt and 10 watts, was very small, difficult to explain, since mutants with less antenna pigments will be expected to greatly outperform wild type. So this suppressing metabolic reaction that divert electron flow away from nitrogenase should increase hydrogen yields. So basically in photofermentation, only for enzyme of interest is nitrogenase. So using nitrogen containing substrates, nitrogenate enzyme is used, which can be extract protons, and by combining protons, they produce hydrogen. So this process is called as photofermentation, and generally it requires low light intensity. So it is a light based type of hydrogen production. What are the advantages? It uses readily available waste streams. So basically, you should have any nitrogen content in the waste material and nearly complete substrate conversion by nitrogen as enzyme. So the efficiency wise, this is very good, but disadvantages are low volumetric rate of production. That means hydrogen produced is very, very, very slow, such that like a tinkering of a water tap. So if you need to fill a tank with water, you should have high water inlet, high flow of water. If it is very low, it will take much more time to fill that particular tank. Similarly here, the rate of production of hydrogen is very low compared to volumetrically. So it requires more and more time, and hence it has got a low efficiency of hydrogen production. As it is an enzymatic process, the rate of enzyme of this enzyme is less. What can be done with this? You can use the strain improvement techniques, what we have discussed in module 1. Some strategies from those in module 1 can be used through metabolic engineering, so that we can replace this nitrogenase with some other uh, enzyme or some other process which will produce hydrogen more than nitrogen. And in future, this improvement seems to be possible due to these antenna mutants. What are those antenna mutants? Is these are the uh, components of nitrogenase enzymes which will help in generation of more hydrogen production. Contrary to light fermentation, dark fermentation is light independent, so it doesn't require any sunlight, and it uses uh, different diverse pathways to produce hydrogen instead of nitrogenase enzyme. But the major problem is only one third substrate is used. So in light fermentation, nearly all substrate is used, but here in dark fermentation, just 75% is the substrate utilization. Remaining, 25%. Remaining 75% substrate uses is getting diverted to form other fermentation products like acetate, butarates, ethanol, like that. So hydrogen here in this dark fermentation seems to be a byproduct of the entire system. So it is not advisable if we rely on hydrogen as a fuel. So preferably we should go for light fermentation, but again disadvantage is low volumetric production. So in terms of growth and survival of microorganism, this is making sense. Along with hydrogen, you will get some other products, and it also favors the growth and survival, and it maintains the redox balance in the fermentation. So these are the complex pathways by which in different steps you will get hydrogens. Like from acetyl CoA, which is a central part of your TCA cycle, you can get ethanol acetate like this. Here you are not getting hydrogen. These are the anaerobic fermentation products. But here when you go from conversion of acetyl CoA to pyruvate, NADH and nickel iron cofactor containing enzymes can produce hydrogen along with their conversion from acetyl CoA to say formate or say to pyruvate or to say any other things. So here in these steps you will see the production of hydrogen, and it is a ATP driven process from sugars to pyruvate. So this first step is ATP driven. Here you get hydrogen. So pre-treatment is same as that of your bioethanol, biogas like that. Sugars again, this dark fermentation is same. As you can see, you are having ethanol here, you can have biodiesel for this, not biodiesel, ethanol and biogas here. Other alcohols, acetic acid maybe here, acetate, But along with that, the same process can be used with certain modification for hydrogen process production also. That's the advantage of dark fermentation. So what are the advantages of this dark fermentation is it can use variety of waste streams, and it can make use of existing infrastructure which is already in use for biogas or bioethanol production. Thus, it has got a simple reactor technology. Non-sterile conditions are acceptable because it's generally a mixture of microorganisms which will produce different byproducts and end products. The disadvantage is being you need to have large amount of byproducts which are raw in nature. Less pure byproducts are acceptable, but then you have to have a large amount, and you have to modify each time when you change the reactor. So there is reactor to reactor variation, and there is very low COD removal. So it puts a burden on your further effluent treatment also. Here, what is expected in future is you can have a two-stage systems which can extract more energy in a cyclical way, so that whatever these hydrogens are generated and these products are generated, whatever effluent what is left over out after recovery of ethanol and hydrogen out from it can be fed back along with combining with sugars generated from pre-treatment, and again the process can be made cyclic to increase its efficiency. That is one of the beneficial thing which can be. So with this, we complete this point of hydrogen production from biology. So we have seen there are two methods of hydrogen production using biological system, light driven and dark driven, or light dependent and light independent. In light dependent, you can have either bacterial photosynthesis or plant type photosynthesis. As plant based photosynthesis, it is out of this syllabus scope. We have seen only bacterial type of photosynthesis where we use enzyme which is called as nitrogenase, which will form from nitrogen waste containing material. It will directly produce hydrogen. It has got certain advantages and disadvantages. Along with that, we saw dark fermentative biohydrogen production, in which we use same similar pathways or similar treatments which we use for bioethanol production and biogas production. We pre-treat the sugars and raw material to form sugars, and then we reuse sugars to form fermentations, and from fermentation you get bioethanol, biogas. Along with that, as a byproduct you are getting hydrogen. So that's it for the point module 3.5, and here it completes the entire module 3.